Today, we're going to pull back the curtain on one of the biggest secrets in all of biology, the Central Dogma. I mean, this is it. This is the story of how a tiny, tiny instruction manual inside every single one of your cells gets read, gets copied, and gets used to build everything that makes you, well, you. So let's start with a fact that is just kind of mind-blowing. If you could take the DNA from just one of your cells and stretch it all the way out, it would be about 6 feet long. And yet all of that fits inside a nucleus so small you need a powerful microscope just to see it. It's the complete blueprint for your entire life, packed with unbelievable efficiency. The big question is, how does your body actually use that blueprint? When you boil it all down, the whole process follows this incredibly simple, elegant flow. Information goes from DNA to RNA and then to protein. That's it. That's the central dogma, and it's the core information highway for pretty much all life on Earth. Just think of it this way. DNA is the master blueprint, locked away for safekeeping. RNA is like a temporary photocopy of just one page. And protein? That's the actual thing you build using the instructions on that copied page. So let's start at the very beginning, copying the master blueprint.
Okay, so first up we have replication. You see, before a cell can divide, whether it's to help you grow or to repair a cut, it has to do something absolutely critical. It has to make a perfect, flawless copy of its entire instruction manual. Every single chromosome has to be duplicated.
Now, this brings up a huge question, right? How on earth does a cell make sure that both of the new cells get an absolutely identical copy of that manual? The answer, it turns out, is a process that is just beautiful in its simplicity. That process is called DNA replication, and the basic idea is just brilliant. You take the famous DNA double helix and you unzip it, pulling the two strands apart right down the middle. Then, each of those original strands becomes a perfect template, like a mold to build a brand new partner. And what do you get at the end? 2 complete, totally identical DNA molecules from the one you started with. Now, there's a technical name for this. It's called the semi-conservative model, and it sounds kind of complicated, but all it really means is that each new DNA molecule conserves 1/2, one strand, from the original parent molecule. This is the secret to its incredible accuracy. By using that original strand as a guide, the cell makes sure there are almost no mistakes. It preserves the blueprint perfectly. To get all this done, the cell has a whole crew of specialized molecular machines called enzymes. Think of it like a construction site. You've got an enzyme called helicase, that's the one that unzips the DNA. Then the main builder, DNA polymerase, comes in and starts adding all the new pieces, and it even proofreads its own work as it goes. And finally, an enzyme called ligase acts like a welder, sealing up all the new pieces into one continuous, perfect strand. And here's where things get really, really clever. Because the 2 DNA strands are like mirror images of each other, they actually have to be copied in different ways. One strand, called the leading strand, gets built in one long, smooth, continuous piece. But the other one, the lagging strand, has to be built backwards in a bunch of small chunks that get stitched together later on. It's a slightly messy but totally ingenious solution to a tricky chemical problem, and it lets the cell copy its DNA at incredible speeds. Okay, so the cell can copy its entire library of blueprints. That's amazing. But, most of the time, a cell doesn't need the whole manual. It just needs one recipe, one instruction, to build one specific thing. And for that, it needs to make a portable, working copy. And this creates a bit of a logistical puzzle. The master blueprint, the DNA, is locked away safely inside the cell's nucleus, where it's protected from damage. But the factories that build the proteins, called ribosomes, are way out in the main part of the cell, the cytoplasm. So how do you get the instructions from the library to the workshop without ever letting the original, priceless book leave the building? The answer? You make a copy. This process is called transcription. Instead of copying all 6 feet of DNA, the cell makes a short, temporary, portable copy of just one single gene. This copy is made of something called messenger RNA, or mRNA. It's exactly like photocopying one page out of a giant encyclopedia to take back to your desk. The star of this show is another enzyme called RNA polymerase. It scans along the DNA until it finds a "start here" signal. It latches on, and then it zips along the gene, building that single-stranded mRNA copy of the code. It's like a molecular scribe, carefully transcribing the message word for word. But wait, we're not quite done. The first copy that RNA polymerase makes is sort of a rough draft. We call it pre-mRNA, and it actually contains a little more information than is needed for the final recipe, so it has to go through an editing phase first. This editing process is all about cutting things out. You see, the parts of the gene that actually have the instructions are called exons. You can think of them as the parts that get expressed. But in between the exons, there are these other non-coding parts called introns, or intervening sequences. The cell's machinery comes in, snips out all the introns, and then splices the exons together to create one clean, final message. And before that finished mRNA message is allowed to leave the nucleus, it gets two last little accessories. A special protective cap is added to the front end, and a long tail of repeating bases is added to the back. It's basically like putting a hard hat and body armor on your little messenger molecule to protect it on its dangerous journey to the factory. All right, our little messenger RNA is now edited, it's protected, and it has left the nucleus. It's headed for the workshop. And this brings us to the grand finale, the final, and you could argue, the most important step, turning that coded message into an actual, physical, functional thing. This final step is called translation, and it all happens out in the cytoplasm, at a tiny little structure called a ribosome. This is it. The ribosome is the cell's protein-making factory, and it's ready to get to work. It just needs to clamp onto that mRNA message and start reading. And translation is the perfect word for it, because that's literally what's happening. The ribosome is translating from one language to a completely different one. It's taking the language of nucleic acids, that sequence of letters in the mRNA, and it's converting it into the language of proteins, which is a specific chain of amino acids. So how does it read this new language? Well, it doesn't read one letter at a time. It reads the message in three-letter words called codons. The ribosome scoots along the mRNA, reading the bases three at a time, and each one of those three-letter codons, like AUG or GCA, is the code for one specific amino acid. It's like a universal dictionary that's used by almost all life on your planet. The whole system works just like a molecular assembly line. The mRNA message slides into the ribosome, which finds the special start codon to get everything going. Then another kind of RNA, called transfer RNA or tRNA, acts like a fleet of tiny delivery trucks. Each tRNA truck carries one specific amino acid, and it knows exactly which codon to deliver it to. As they arrive, the ribosome links the amino acids together, one after another, building this long chain until, bam, it hits a stop codon. That's the signal that the protein is finished and it's released to go do its job. So there you have it, DNA to RNA to protein, replication, transcription, translation. It's a simple one-way flow of information that acts as the fundamental operating system for life. It's how a static, unchanging code stored in DNA can create the dynamic, complex, and beautiful reality of a living thing. And look, this isn't just some abstract diagram in a textbook. This is happening inside you right now, billions and billions of times every single second. The color of your hair, the enzymes that are digesting your food, the antibodies fighting off a cold. Every single one of those is a protein that was built using this exact process. This elegant, precise, and frankly beautiful system builds everything that makes you who you are. But it makes you wonder, doesn't it? In a code that's billions of letters long, running constantly, what happens when there's a mistake? What happens if there's just a single typo in the code? Well, that question, that changes everything.